7.5 has no new material. It's just an opportunity for you to check yourself. Please make sure you've watched the BioFlix video on cellular respiration, but you should also be reviewing the slides and any additional videos until you feel comfortable. It's not very useful to check yourself if you haven't actually gone through and studied, because then you just say that, well, I'll just study later. The purpose of this video isn't about that. This is about taking an opportunity to work with the material and then using these questions to see how well you understand it. You're going to finish out the last of Socrative, and you only get one go at it, which means you only get one opportunity to test yourself. So please make sure that you give it a try before you stop and do these questions. Once done, finish the video to review those answers. So don't follow along with me. Go ahead and finish Socrative and then come back again and go through these answers to see how you did. So pause here, do the last 9 questions in Socrative, and then come back. The first question asks you to take a look at the forces that are being built by the electron transport chain and to understand the differences between NADH and FADH2. Recognize that NADH and FADH2 both donate 2 electrons. So there's nothing different about them in terms of the number of electrons that are being donated. And yet FADH2 results in the production of less ATP, 1.5, than NADH, 2.5 ATP. And what is the reason for that difference? Well, if you take a look at the path of the electrons that are being donated, NADH donates electrons here in complex 1. So they're going to pass through complex 1, complex 3, and complex 4 on their way to being donated to oxygen. But the electrons from FADH2 get donated here in complex 2. And since they don't go backwards, they get donated or pass through 3 and 4 on their way to oxygen. So they only go through 2 electron carrier molecules. So each one of these electron transport molecules is going to be responsible for pumping a proton. And so FADH2's electrons are actually going to be donated later in this chain, which means that they're going to pass through fewer of these electron transport complexes, and so fewer protons are going to be pumped. That means it contributes less to the proton motive force, and so is responsible for the production of less ATP. The reason why the cell does this is because the electrons in FADH2 are going to start out as lower free energy. The molecule that donates electrons to FADH2 is actually slightly different than all the molecules that are responsible for donating electrons to NADH. And these are lower free energy electrons, and the molecule FADH2 just isn't as high in free energy as NADH. But it's better than not accepting any electrons. And so the cell, at some point, evolved to be able to utilize FADH2 in this particular process. So what would happen if to the rate of ATP synthesis if we were to replace oxygen here at the end with a molecule of sulfur? Recognize that sulfur is going to have a lower electronegativity than oxygen does. And since that electronegativity determines the rate at which these low energy electrons get donated, that also means that the rate at which these electrons get donated is less. Since that rate has decreased, the production of that electron has also decreased. And since we're still doing an electron transport chain here, this is still a process of cellular respiration. But since we don't have oxygen, this would be a process of anaerobic respiration. Here's another opportunity for you to be able to test yourself. And you're certainly welcome to pause the video here or extract this video. You're going to be doing it in recitation. So see if you can name the steps and count the molecules and understand the production of ATP at each one of these steps. So which of the following about glycolysis is true? Well, glycolysis does not create ATP by oxidative phosphorylation. It does so by substrate level phosphorylation. And oxygen is not necessary for any stage of glycolysis. It produces not the most ATP by any means. It produces very little. As a matter of fact, we net only 2 ATP, and it does not synthesize any lipids, but it does occur primarily in the cytoplasm. When we look at the citric acid cycle is not dealing with glucose. This is glycolysis. And it does make ATP, but again, not the most ATP. Most ATP is going to be made by chemiosmosis. It does not occur in the cytoplasm. Glycolysis occurs in the cytoplasm, but the oxidation of pyruvate has transitioned this molecule of pyruvate into the mitochondria when it produced the acetyl-CoA. And once again, citric acid cycle does not require oxygen, but it does produce 2 ATP through substrate level phosphorylation. When we take a look at that energetic electron being taken from glucose or the broken down products of glucose, we're utilizing the energy to be able to transport protons. When you look at this diagram here, it's important to note that the protons are what's being pumped. We don't pump electrons, we don't pump NAD+, we don't transport any of them. This is all about the fact that we're pumping protons. It's also important to note that the space in which those protons are pumped is the intermembrane space, not into the matrix. So ATP synthase at the inner mitochondrial membrane makes ATP by which of these methods? This would be the facilitated diffusion of H+. And it's going to utilize the energy from this diffusion, the ability of this H to be moving back down its concentration gradient from an area that has high H+ to an area with low. And the energy which is released from this is going to be the energy that catalyzes the production of ATP. Oxidative phosphorylation is achieved through which combination? Well, we have glycolysis, but that's not part of our oxidative phosphorylation. So yes, we do need to utilize glycolysis, but not here. An electrochemical gradient is necessary, but it's not enough to have it on its own. Oxidative phosphorylation also does not include chemiosmosis. Phosphorylated intermediate in a kinase is going to be energy coupling, not the process here. And the splitting of oxygen molecules is not related. Oxidative phosphorylation is the electron transport chain combined with chemiosmosis. So which of these describes anaerobic cellular respiration? Well, glycolysis only and no electron transport chain. This is fermentation. The electron transport chain, where oxygen serves as the terminal electron acceptor, is going to be aerobic cellular respiration. And this is asking for anaerobic cellular respiration. So the electron transport chain where sulfate serves as our terminal electron acceptor would be an example of an anaerobic process.